can.
Okay. Okay, so our group did protein synthesis, and because of our food choices, we can't really wrap a churro into a double helix. So what we did was we modeled a mini churro into the shape that DNA is originally supposed to be in. Okay, so the first step in protein synthesis is that this little guy, it's like a little tiny piece of burnt biscotti. It's the RNA polymerase, and what it does is that it scans the DNA until it finds a certain sequence of nucleotides, DNA nucleotides, which is called a promoter. And what it does is once it finds a promoter, it binds to the promoter, and it unwinds the DNA, which breaks all the hydrogen bonds between the DNA like this. So if we zoom into our DNA strand, this is what our DNA would look like. This is the big one.
Okay, so this is the RNA polymerase, and what the RNA polymerase does is it finds RNA nucleotides in the nucleus, and it bonds them hydrogenically, the nitrogenous bases hydrogenically, and then it bonds the RNA nucleotides covalently. The differences between the DNA nucleotides and the RNA nucleotides are RNA has uracil instead of thymine, and RNA has ribose instead of deoxyribose.
Oh, and before we move any further, just for like clarification, the RNA polymerase finds RNA nucleotides that are complementary to the bases on the DNA strand, just like it would on DNA replication. But as David mentioned before, it instead of thymine, it uses the base, nitrogenous base, uracil. And it does this one, oh yeah, here you go, keep going. It does this one, also, these, this green frosting represents, and the chocolate coating, it represents the differences in the sugars, the 5 carbon sugars in the RNA and the DNA. So this chocolate would represent the sugar deoxyribose in DNA nucleotides, and the green frosting would represent the sugar ribose in RNA nucleotides. And so what it does is the RNA
polymerase eventually forms this whole strand of mRNA, and the mRNA is hydrogenically bonded to the DNA at first, and then it is separated and then moved into outside of the nucleus into the cytosol.
And what it does is that the RNA polymerase knows when to stop transcribing the code from the DNA strand to the mRNA strand because of a termination signal, which is another sequence, like the promoter is another sequence of DNA nucleotides. And basically it says, since mRNA is only the length of a gene, the termination signal basically states that, okay, the gene sequence is over. So the RNA polymerase lets go of the newly made mRNA, and it lets go of the DNA that it was transcribing the mRNA from, and it lets the DNA wind up again, forming hydrogen bonds between the nitrogenous bases like it was before. Okay.
Okay, so the mRNA is moved outside of the nucleus
Through one of these nuclear pores.
Through the pores in the nuclear envelope, and then it binds to a ribosome.
Okay, sorry. And what happens is that the ribosome detects, in the mRNA, each triplet of RNA nucleotides is called a codon, and there's one specific start codon to start the process of protein synthesis in the ribosome, and
it's AUG, and what AUG is, these are called the anticodons. The anticodons give the genetic code for the amino acid, which is methionine, and the anticodons and the codons hydrogenically bond.
And the anticodons on a strand of tRNA and the corresponding codons on mRNA, they're complementary to each other. Okay, so after the first tRNA comes in and bonds with the start codon in the mRNA strand. The ribosome, or it can be the mRNA strand, moves along the ribosome to make room for the next tRNA molecule, which will bind to another codon. This particular codon happens to code for the amino acid glycine. Okay, David, do you want to explain what happens after that?
After glycine bonds, the tRNA, the methionine forms a peptide bond with the glycine, and then the tRNA leaves the ribosome and goes to go pick up more amino acids.
Of the amino acid methionine. And this process keeps continuing, so this would move, and then the next, it's already there, carrying another amino acid. And then these two amino acids, the glycine and the serine, would form a peptide bond. Here it is. Modeled by the toothpick in between the two of them. And then, now that the tRNA molecule that came before it knows that it did its job, it flies away to find another amino acid. And finally, sorry, we made a really long strand. Another tRNA molecule comes in, finds its appropriate codon, and then these two amino acids bond, and this tRNA goes away. And now, what happens next is something very special.
After the hydrogen bonds form, the next tRNA comes.
No. Because, this is supposed to be AUG. Okay, well,
The next section of code is UAA, which stands for the stop codon, which basically means that it tells the ribosome to stop doing transcription, protein synthesis, and the tRNA goes away.
and these
Then the ribosome moves away, and then the mRNA breaks up into nucleotides and goes into the nucleus to be used again.
And most importantly, these amino acids, they fold up to become the protein that they were meant to be, because the reason why the shape of proteins are so critical to its function is because proteins a lot of times often act as enzymes, and as we learned a few chapters before, a protein has to be a specific shape for it to catalyze a certain reaction, and that's protein synthesis.